Hello lovelies, in this video the brilliant Dr. Edwards is going to take you through the development of drugs, the stages drug development goes through and what we're looking for before a drug actually gets to market. This is everything you need to know for your A-level biology.
Development of drugs. So although we currently have a lot of medicines and drugs available to treat patients, sources of new medicines are still needed for various reasons. New diseases are constantly emerging, and obviously we saw this with COVID in the last few years. There are still some untreatable or uncurable diseases, and obviously some of these are genetic and inherited, but there are some others that don't respond particularly well to treatment or don't have a specific treatment. And many bacteria have now evolved resistance to certain antibiotics. So MRSA and C. difficile are two examples of bacteria which now you really can't treat them with most antibiotics because they are resistant. So how could we potentially discover some new drugs or sources of medicine? Accidental findings plus collaboration from loads of different people. If we do find something, which is how we ended up having penicillin. So it was accidentally discovered by Alexander Fleming, but only through working and collaborating with other scientists were they able to purify it and get it to the antibiotic that we know today. So this could happen again through various trials of research. We could accidentally discover something. Using traditional remedies as a start point and then extracting and synthesizing their chemicals. So for example, morphine, which comes from poppies, that drug that was originally used and kind of extracts of poppies were used to create opium and other drugs that were used as traditional remedies a long time before we had modern medicine. So finding out plants that were used for various treatments in the olden days and then trying to extract and synthesize whatever chemical it was that has the active ingredient in order to make some drugs. And observing wildlife using medicinal plants. So there are various animals that will be seen using plants as types of medicine. So for example, rubbing the essential oils of various leaves of plants onto their skin or fur to try and prevent insect bites. So being able to observe these in nature and then maybe trace back what plant they were using and see if that plant could have helpful properties for us and could be useful for a drug is also a method we could use to discover new medicine. So what current research and development techniques are we using to try and help get more information that can help lead us to new sources of medicine? So one is to use sort of tropical plants as the basis for our research, analysing kind of rainforests and finding new plant species and new tropical plant species that wouldn't have necessarily been used or written down in old remedy books and from records we've got from Western countries. But if we go south of the equator and find where there's really diverse species, there's a chance that they could contain chemicals we've not discovered before that could be used as a new medicine or a drug. So using chemical fingerprinting technology to screen natural chemicals in these plants for their kind of potential medicinal properties. This is important, and also therefore it's important that we conserve biodiversity, especially in tropical areas, because we know that biodiversity is actually reducing there faster than in other places. And this is because obviously we don't want to lose potential resources for future medicines that we haven't even potentially discovered yet because they get made extinct due to human activity or something like that, due to the loss of biodiversity. So that's one thing that we need to kind of focus on as well, and they need to be done together. Researching and improving our understanding into how microorganisms cause disease is also really important, because then these mechanisms can be the target for drugs to try and prevent, say, for example, their entry into cells or their ability to replicate within the body. Once we understand how the mechanism works and what they need to use it, then we can start targeting drugs to inhibit enzymes or bind to receptors and therefore stop them carrying out these processes. And finally, synthetic biology. So developing new molecules, synthetic production of biological style molecules. So it could be enzymes or proteins or molecules that are the same shape and have the same activity as various proteins in the body. And we could do this by genetically modifying organisms using new nanotechnology to create very, very tiny molecules. And this is going to help us to kind of actually promote and produce new drugs that maybe would be really difficult to extract or would be really expensive to extract, or using the actual biological molecule is too difficult, or it's really rare. So from a rare plant or something, we can actually make these molecules synthetically in order for them to work. So where could we be looking in terms of the future of medicine? Well, Personalized medicine is kind of the next step in how we improve medicine for everybody. So personalized medicine can be used to target different dosages or compounds, so drugs, as medicine depending on the genetic makeup of the person. So the technology is in its infancy, but it has the potential to revolutionize the effectiveness of a wide range of different medicines for different conditions. And it would obviously rely on us sequencing people's DNA to actually know their genome, understand their genome, and then maybe isolate targets for various diseases and drug therapies. So a good example of this is some breast cancer treatments, and these are targeted at specific mutations. So screening people to make sure that they have the specific mutation before giving them the breast cancer drug means that they've got a better chance of that drug actually working. So say, for example, a load of people have lung cancer. It's obviously one of the major cancers. We take blood samples from patients with lung cancer. We are going to take part of their DNA and we're going to amplify it. So we're going to isolate genes that we know could be there for lung cancer. And then we're going to amplify those genes using the polymerase chain reaction to get more copies of that DNA. Then we could sequence that DNA to see what genes that they have and if they have the genes for the different types of lung cancer, or we could use a gene probe to find specific genes within their DNA sample. And then each individual person will find out what type of lung cancer they have in terms of their genetics, what mutation is causing their lung cancer. So they will get individual targeted therapies for each person, depending on what mutation they have and what drug we think will best treat their type of lung cancer. Obviously, we're not here yet. We're not at this stage yet. And sequencing people's DNA and really drilling down into what mutations they have and understanding what mutations cause disease is something we don't actually know yet. But this is where the future of medicine is headed. Antibiotics. So since the discovery of penicillin, antibiotics have been used to treat bacterial diseases by killing the bacterial pathogen or preventing it from replicating inside the body. So antibiotics are drugs that do directly affect a pathogen, but only bacteria. Since 1928, the use of these drugs has saved many lives, and it has reduced the number of deaths from bacterial diseases across the world. This means it has increased our life expectancy. So how long people have actually lived for as a maximum age or an average age has increased since we had antibiotics, because so many people would normally have just died from bacterial infections. However, their effect is kind of wearing off and they're becoming less and less effective because the bacteria are evolving to be resistant to these antibiotics. The main example we use is MRSA. This is a bacterial pathogen that is now known to be resistant to most antibiotics, which means if you get infected with MRSA, there isn't much we can do to help treat you. We need to be able to explain antibiotic resistance and how it happens. So there's variation in a bacteria population always, and this is due to random genetic mutations in the population. This means that there are some bacteria in every population which will be resistant to the antibiotics you're going to use to treat them. So in this case, it's my pink bacteria in this colony. So when we add the antibiotic, majority of the non-resistant bacteria will die and the resistant bacteria will survive. They will then be able to reproduce and they will pass on their resistance genes or their resistance mutation to their offspring. This increases the number of resistant bacteria in the population until the majority of the population is made up of resistant bacteria. So if we were to treat this population with the antibiotic again, or the same antibiotic, then nothing would happen and the majority of the population would survive. So gonorrhea is another example where there are a couple of strains of gonorrhea that are also now antibiotic resistant. So we need to be able to give some examples of ways we can try and prevent antibiotic resistance from developing further. The first one is to reduce our overuse of antibiotics when they're not needed. So doctors should not be prescribing them for viral infections because obviously if you have a virus, taking antibiotics will not help cure your virus. They will not affect the virus at all because they only kill bacteria. And what they can do is make some of your bacteria that live inside your body, especially the ones in your gut, resistant to antibiotics. The second step is to make sure that anyone who's been given a course of antibiotics takes the full course. That means you finish every single tablet in the packet as your doctor instructed, and you don't just give up taking them as soon as you start to feel better. This helps to reduce the number of bacteria that are surviving because if you keep taking the antibiotics and keep taking them, You're going to try and guarantee that you'll kill the maximum amount of the bacteria, not leave any that could survive, and then reproduce. We also need to reduce the standard use of antibiotics in farm animals. So often antibiotics are put in animal food, or they're just given to animals regularly because it's to try and prevent them from getting sick with diseases. But actually that's dangerous because it could mean that we could create a bacterial-resistant colony of bacteria or strain of bacteria, and these could then be passed on to humans.
Ouch! This is why in some videos I'll explain scratches.